purpose of DNA is to store and transmit genetic information. Chromosomes, where DNA is stored, can come in many different shapes and sizes. What's important is that DNA is safely stored with a stable foundation. In prokaryotes, chromosomes are circular, and eukaryotic cells have linear and multiple chromosomes. Both types of cells may contain plasmids, small circular double-stranded DNA molecules that replicate independently.
DNA is a double-stranded molecule that is made up of nucleotides. A nucleotide is made up of a 5-carbon sugar, a phosphate group, and a nitrogenous base. These nitrogenous bases are adenine, thymine, cytosine, and guanine, with a person for RNA.
Chromosomes contain genes that store specific genetic information. This is how genetic information was passed to daughter cells in meiosis, and how genetic diversity is accomplished. The random chance of these chromosomes being in the specific daughter cells is how genetic diversity is accomplished. Furthermore, events in meiosis such as crossing over, fertilization, mutations, and etc. increase diversity as well.
Adenine binds to thymine or uracil with a double bond, while cytosine bonds to guanine with a triple bond. Adenine and guanine are purines, which are double ring. Thymine, uracil, and cytosine are thymines, which are single ring. DNA replication is a holistic process that has many vital steps. Enzymes are primary helpers that make the DNA replication process possible. DNA should also be capable of differential expression so that the various parts of the organism may acquire specific form, structure, and function in spite of having the same genetic material.
DNA replication is an essential function of the DNA in order to make new cells. It starts by unwinding itself, and then DNA helicase helps to disrupt the hydrogen bonds that hold the base pairs of each strand of DNA together. This is a reaction that requires energy, and following this disruption, the DNA is then separated into two strands. It's similar to unzipping a zipper, where it starts at multiple points of the DNA and cuts both sides of the double-stranded DNA and makes its way through in order to have two templates. Also, why DNA is semi-conservative, because the original strand is now two templates for two sets of DNA. It's also important to know that helicase creates replication bubbles, which means that all the DNA in the cell is being worked on by multiple helicase, which creates a pocket where replication is happening. This basically saves time and makes the process more efficient, especially when there are billions of base pairs. Also, for SSP proteins, single-stranded binding proteins stabilize the unwound proteins and keeps the parental strands from rebinding.
Primase is a DNA-dependent RNA that serves a vital role in the process of DNA replication. DNA primase initiates the addition of foreign and new nitrogen-heavy bases to the single-stranded DNA after the actions of helicase. Primase adds a strip of RNA to the single-stranded DNA referred to as a primer. Primer serves as the foundation of DNA synthesis and functions as the starting point. The strip of RNA serves as an arrow guiding DNA polymerase. The leading strand only has one primer so that the DNA polymerase knows where to go. The primase keeps on placing primers for the lagging strand so that DNA polymerase can keep building up on the 3' strand. The reason why the lagging strand needs multiple primers is because DNA replicated from 5 to 3 direction, which can cause discontinuous synthesis of the DNA in the lagging strand, which is 3 to 5. If primer is not added or somehow blocked, DNA replication would not proceed and result in failure.
DNA polymerase III is an enzyme that creates DNA molecules. They do this through the process of assembling nucleotides in the direction that the primers tell them to. Adenine goes with thymine and guanine goes with cytosine. Polymerase III adds 2 hydrogen bonds between adenine and thymine and 3 hydrogen bonds between guanine and cytosine. Polymerase is essential to DNA replication, and they usually work in pairs to read the existing DNA and create 2 identical DNA strands. When making the new half for the lagging strand, DNA polymerase III, which depends on the primers to know where to build, will make discontinuous fragments. These fragments are called Okazaki fragments, which are bridges that are not fully built, and this is because the lagging strand is 3' to 5', but DNA is only built in the 5' to 3' direction, so there is a lag while helicase cuts more bonds. Also, DNA polymerase 1 gets rid of the primers and replaces them with the DNA.
Ligase plays an important role in DNA replication because it connects the old strands with the new strands, and ligase can even act as a glue that sticks the separated Okazaki fragments to the DNA that polymerase 1 replaced the primers with to make a continuous strand of DNA. As the ligase connects the strands, the DNA becomes two continuous strands. Without the ligase, the DNA can't be two continuous strands and will break down. This would mean that bigger processes such as mitosis and meiosis would not happen, and new cells, if the damaged DNA is still carried over, wouldn't be able to do certain actions. The damaged DNA could even result in dead cells or tumors if the damaged DNA kept getting passed on.
Topoisomerase is the enzyme that releases tension and releases mechanical energy. You can think of the DNA strand as a string that coils as the helicase cuts, and the topoisomerase is the tension reliever that pulls the string and entangles it. Topoisomerase makes helicase more efficient and allows helicase to unzip more efficiently. If the DNA is too gently wound, it's hard for helicase to unzip and will cause some DNA to be lost. If the DNA is relaxed, it's easier for helicase to separate the hydrogen bonds and open up the two strands. All these processes, by the way, from the very beginning, from the unwinding of DNA to the creation of new strands, keep happening simultaneously.